In March of 2026, a team of geneticists opened a set of linen fibers that had been sitting in storage since a single night in October of 1978, and what they pulled out of those threads has already reached the Vatican, reignited a 600-year argument, and forced historians, chemists, and believers alike to ask the same question all over again. Not whether this cloth is a fake, not whether it is real. Something narrower, stranger, and in some ways more interesting than either of those two options. What exactly has this piece of fabric been touching, breathing, and absorbing for the last several centuries of its existence? And why does the answer refuse to line up with the simple story anyone wants to tell about it? The object at the center of all this is the Shroud of Turin, a herringbone weave linen cloth roughly 4.5 meters long, kept today inside Turin Cathedral in northern Italy. It carries a faint, straw-colored image of a man, front and back, head to head, as though the cloth had been laid beneath him and folded back over his head. The figure shows wounds at the wrists, puncture marks around the scalp, lacerations across the back and shoulders consistent with scourging, and a wound in the side. For believers, this is the burial cloth of Jesus of Nazareth. For skeptics, it is one of the most successful forgeries in European history. What makes the object genuinely unusual is that, unlike almost every other contested relic in the world, it has spent more than a century being handed directly to scientists, over and over again, and asked to speak for itself. That process effectively began in 1898, when an Italian lawyer and amateur photographer named Secondo Pia was given permission to photograph the cloth during a public exposition. When he developed his glass negative plates in a darkroom that night, he found something nobody expected. The faint, almost illegible image on the cloth reversed into a strikingly detailed, anatomically coherent picture of a human face and body. The kind of clarity you would expect from a photographic negative, not from a painting. That single accident, his eye, had pulled the shroud out of the world of pilgrimage and devotion and dropped it into the world of forensic science, where it has remained under one microscope or another ever since. That shift accelerated in 1978, when a team of American scientists calling themselves the Shroud of Turin Research Project, usually shortened to STURP, was granted 5 consecutive days of direct, round-the-clock access to the cloth inside the royal palace in Turin. This was an unusually generous arrangement for a religious relic, and it came with real scientific weight behind it. The team included physicists, chemists, and imaging specialists from institutions such as Los Alamos National Laboratory in the air, Force Academy, and they brought with them X-ray equipment, ultraviolet and infrared photography, microscopy, and adhesive tape sampling designed to lift microscopic particles directly from the weave without cutting the fabric itself. It was during this exact 5-day window on the night of October 8th and 9th that the samples now at the center of the 2026 genetics study were originally collected. STURP's own conclusions, published in the years that followed, stated that the group could not identify any pigment, dye, paint, or other artistic medium consistent with the image, and could not, using the technology available at the time, explain a more cautious statement than it is often given credit for. STURP explicitly did not conclude the shroud was authentic. It concluded that its own instruments could not explain the image through any known forgery technique available at the time, which is a narrower and more defensible claim, even if it is frequently overstated in popular retellings. Decades of study by chemists, textile experts, and forensic pathologists have confirmed a few things that are not seriously disputed. The image sits only on the very topmost fibers of the linen threads, a discoloration measured in a few hundred nanometers of depth, with no evidence of brush strokes, no binder, and no pigment consistent with any known medieval painting technique. Blood-like stains on the cloth have tested positive for the presence of blood components in multiple independent laboratory analyses, and the flow patterns of those stains are, according to several forensic reviewers, consistent with wounds inflicted on a body in specific postures, rather than patterns a painter working from imagination would likely reproduce by accident. None of this proves who the man in the image was. It simply establishes that whatever produced the image was not obviously an act of medieval painting, which is exactly why the object has remained a live scientific question instead of a settled historical footnote. The most famous scientific chapter in the Shroud story came in 1988, when 3 independent radiocarbon dating laboratories at the University of Arizona, the University of Oxford, and ETH Zurich tested small samples cut from one corner of the cloth. All 3 labs returned strikingly consistent results, dating the linen to somewhere between the years 1260 and 1390. That window lines up neatly with the earliest confirmed historical record of the shroud's existence, a badge sold to pilgrims at Lire, France, dated between roughly 1350 and 1418, which shows the same double image and is generally accès, PT'd by historians on both sides of the debate as genuine evidence of the shroud's presence in medieval France. For many scientists, that overlap between radiocarbon dating and documented history settled the matter. The shroud, by this account, is a 14th century object, and the search for who painted it or produced it becomes a question of medieval technique rather than ancient burial practice. That conclusion did not go unchallenged. Chemist Raymond Rogers, who had worked directly with the shroud as part of the 1978 scientific research project, argued in the mid-2000s that the corner sampled for radiocarbon testing showed signs of having been rewoven at some point in its history, potentially during restoration work following a documented fire. If the tested threads were partially medieval repair material rather than the original fabric, Rogers argued, the radiocarbon result would reflect the age of the patch, not the cloth. This argument gained real traction in popular discussion of the shroud and is still cited frequently today, but it has not gone unanswered. A 2026 textile analysis published in the journal NPJ Heritage Science examined the specific fragments and surrounding fibers in detail and reported that the samples were consistent with the shroud's main body and showed no clear evidence of the kind of contamination or rewoven material Rogers proposed. As things currently stand, radiocarbon dating remains the single most rigorous, independently replicated test ever performed directly on the shroud's own fabric, and no later study has produced a comparably strong, peer-reviewed result overturning it. That does not end the conversation. It simply means the burden of proof for an earlier date still rests with the challengers, not with the original test. Which brings us to what actually happened in 2026, and why it is different from anything that came before it. Genetic research on the shroud is not new. In 2015, a team led by geneticist Gianni Barcaccia at the University of Padua published the first major DNA analysis of dust vacuumed from the cloth's surface, identifying plant DNA from species. Scattered across the globe in human mitochondrial DNA, suggesting a wide range of geographic origins among the people who had come into contact with the fabric. That study made headlines at the time, and it also drew criticism, because the techniques used were limited to amplifying small, targeted stretches of DNA, a method vulnerable to contamination and difficult to interpret with real statistical confidence. The 2026 study, again led by Barcaccia, this time working with geneticist Alessandro Achilli of the University of Pavia, and an international team including forensic side different approach. Instead of targeting specific genetic markers, the team used metagenomic sequencing, a method that reads essentially every fragment of genetic material present in a sample at once, human, plant, animal, fungal, and microbial, without deciding in advance what it expects to find. They worked with linen strands taken from the official 1978 sample collection, gathered on the night of October 8th and 9th of that year by Professor Pierluigi Baima Bologna as part of the landmark scientific investigation known as the Shroud of Turin research project. The new study was first posted as a preprint on the biology server bioRxiv in March of 2026, and by July of that year it had passed peer review and was formally published in the journal Scientific Reports. 10 days after the preprint appeared, Vatican News reported on the findings, and the story spread rapidly through international media in the days following Easter, a piece of timing that struck many observers as remarkable, whatever meaning you choose to attach to it. What the metagenomic data actually showed is more complicated, and in some ways more honest, than the version of the story that spread across headlines and video platforms in the weeks that followed. On the human side, the researchers identified several distinct mitochondrial DNA lineages within the samples. One of them, labeled K1AB1A, matched precisely the known mitochondrial profile of Professor Baima Bologna, the scientist who personally collected the original samples back in 1978. That detail matters enormously, because it demonstrates directly and concretely that at least some of the human genetic material on the cloth comes from modern researchers who have handled it, not from anyone connected to its ancient or medieval history. A second lineage, H2A2, corresponds to the reference sequence used as the baseline for virtually all human mitochondrial comparisons worldwide, meaning it is so common across so many populations that its presence tells researchers very little about geographic origin on its own. A third lineage, H1B, is broadly common across Western Eurasia, consistent with centuries of handling by European clergy, conservators, and pilgrims. The lineage that generated the most public excitement was H33, a haplogroup that is comparatively rare in Western Europe, but notably more frequent in the Near East, and specifically among the Druze, an ethno-religious community with historical roots across Syria, Lebanon, Israel, and Jordan. It is worth pausing on exactly what that finding does and does not tell us. Finding a Near Eastern-associated mitochondrial lineage on the cloth is genuinely interesting, and it echoes signals reported in the earlier 2015 study. But mitochondrial DNA of this kind can persist on an object for centuries, through casual contact, and the Catholic Church has never disputed that the shroud has had contact with people of Near Eastern, North African, and broader Mediterranean background throughout its documented history in Europe, whether through pilgrims, clergy of Eastern Christian background, or, according to some scholars, an earlier period of display in Edessa, a city in what is now southeastern Turkey. before its documented arrival in France. The researchers themselves were careful and explicit on this point. Their published conclusions do not claim the DNA proves anything about the shroud's authenticity or its age. What it demonstrates is a genuinely diverse mosaic of human contact stretched across a very long and geographically wide history. Exactly the kind of biological signature you would expect from an object that has been touched, kissed, folded, and displayed by enormous numbers of people across many centuries and quite possibly several countries. A separate and, from a historian's perspective, arguably more revealing part of the 2026 study focused on plant, animal, and microbial DNA. Here, the picture becomes genuinely striking, though not in the direction most viral headlines suggested. The researchers detected DNA from at least 19 distinct plant species. Carrot accounted for roughly a third of all plant DNA identified. Wheat, rye, various melons, peppers, tomatoes, and potatoes also appeared, alongside almonds, walnuts, and several wild grasses. Crucially, some of these species, including maize, tomatoes, potatoes, and peanuts, are native to the Americas and were entirely unknown in Europe, Africa, or Asia before Christopher Columbus's voyages beginning in 1492. Their presence on the cloth is not a mystery pointing toward ancient origins. It is the opposite. It is direct genetic proof that the shroud has continued to accumulate contamination well into the modern era, long after any medieval or ancient date under discussion, almost certainly through storage, handling, cleaning, and proximity to ordinary household and agricultural environments in the centuries following the European colonization of the Americas. The animal DNA told a similar story. Dogs and cats together accounted for close to 44% of all animal genetic material recovered, alongside traces from chickens, cattle, goats, sheep, pigs, horses, deer, and rabbits, along with incidental traces of skin mites, ticks, and even fish. None of this suggests anything mystical. It suggests exactly what you would expect from a piece of fabric that has spent centuries being folded, stored, transported, and displayed in ordinary European households, churches, and travel conditions, surrounded by domestic animals, pests, and food. Perhaps the most unexpectedly charming detail in the entire study was the discovery of DNA from Mediterranean red coral, Corallium rubrum. Barcaccia and his colleagues suggested this likely reflects contact with coral crucifixes, rosaries, or reliquary ornaments, a common devotional material in Catholic Europe for centuries, rather than any connection to the cloth's ancient history. Then there is the detail that captured the most public attention, and understandably so. The reconstructed microbiome of the shroud included, alongside typical human skin bacteria and various molds and fungi consistent with long-term storage, a community of archaea specifically adapted to environments of high salinity. Salt-tolerant microorganisms of this kind are found in a range of extreme environments around the world. The researchers themselves framed this finding cautiously, noting it as one more data point consistent with the cloth's long and varied preservation history, rather than definitive proof of any single location. It is true that highly saline environments, including places like the Dead Sea region, support these kinds of organisms. It is also true that salt has been used in textile processing, leather curing, and fabric preservation across many cultures and centuries, and that salt-adapted microbes can establish themselves wherever suitable conditions exist over time, including through contact with saline preservation materials used in later restoration or storage. The honest scientific reading is that this finding adds one more layer to an already complicated environmental history, not that it locates the shroud definitively at any particular ancient burial site. The study also examined the shroud's silver reliquary, the ornate case in which the cloth has been stored and transported for centuries. Radiocarbon dating of two separate textile residuals found within that reliquary return dates between 1451 and 1800, and the researchers linked these more precisely to documented repair interventions carried out on the cloth in the years 1534 and 1694, following a serious fire that damaged the shroud in Chambéry in 1532. This detail is significant because it independently confirms something historians already knew from written records, that the shroud underwent a real physical repair work using contemporaneous 16th and 17th century thread, work that would naturally have introduced additional textile material and biological contamination, completely unrelated to the original cloth's age. Reaction to the study split along fairly predictable lines. Some shroud researchers and Catholic commentators highlighted the Near Eastern mitochondrial signal and the Druze-associated haplogroup as new supporting evidence for an authentic ancient origin. Historians who study the shroud more skeptically, including scholars like Andrea Nicolotti, who has written extensively and critically about the object's medieval history, pushed back particularly against interpretations suggesting the linen itself might have originated in India based on certain rare lineages, calling that leap unsupported by the broader body of textile and historical evidence, and reaffirming that the 1988 radiocarbon date still represents the strongest direct physical dating evidence available. Barkaccia and his co-authors, to their credit, were notably restrained in their own published conclusions, repeatedly stating that their metagenomic findings illuminate the cloth's biological and environmental history rather than settling any question of age or authenticity. That is an important distinction, and it is the one that tends to get lost the moment a headline writer or video script needs a dramatic hook. It is also worth noting how this study was received within the scientific press itself, as opposed to general news coverage. Outlets covering the chemistry and genetics side of the story, including specialist publications like Chemistry World, framed the results primarily as a demonstration of what modern forensic genomics and metagenomic sequencing can now accomplish on a contaminated, centuries-old textile, rather than as a breakthrough in the authenticity debate itself. That framing matters. The real achievement of the 2026 study is methodological. It shows that researchers can now reconstruct a detailed, layered biological history from an object that has been touched by countless hands across hundreds of years, separating out modern contamination, post-Columbian agricultural contact, 16th and 17th century repair work, and older, harder-to-place genetic signals, all from the same small set of fibers. That is a genuinely significant advance in forensic science generally, independent of whatever conclusion any individual viewer wants to draw about the shroud itself. The shroud is not the only relic caught up in this ongoing forensic investigation. A separate, smaller cloth, known as the Sudarium of Oviedo, has been kept in northern Spain for over 1,000 years, with a documented history that, according to some scholars, can be traced back considerably, further than the shroud's own confirmed record in Europe. Unlike the shroud, the Sudarium carries no image at all, only bloodstains and fluid marks consistent with having been wrapped around the head of a deceased man, a practice described in first-century Jewish burial customs and referenced in the biblical account of Jesus' burial. Forensic researchers, most notably Spanish physician Alfonso Sanchez Hermosilla and Italian criminologist Giulio Fanti's collaborators, have compared blood flow, patterns, wound placement around the nose and beard, and blood type markers between the two cloths, and several independent papers have reported meaningful overlap consistent with both cloths having covered the same or a similarly wounded individual. This research remains genuinely contested. Bloodstain pattern analysis on textiles. This old carries real uncertainty, and centuries of independent handling make definitive proof extremely difficult, but it represents a real, ongoing area of forensic study rather than pure speculation. What makes the Sudarium a particularly interesting piece of this puzzle is its documented paper trail. Spanish records place the cloth in the region for well over 1,000 years, with some historians tracing references to it back to the 7th century, centuries before the shroud's earliest confirmed appearance in France. If the two cloths really did cover the same individual, as several forensic teams have argued based on the geometry and overlap of the stains, that would imply the shroud existed, in some form, well before the 1260 to 1390 window established by radiocarbon testing. That is precisely why critics of the shroud's authenticity tend to treat the Sudarium comparisons with particular caution, since accepting that link creates real tension with the radiocarbon evidence that many scientists consider the most solid data point in the entire debate. Proponents counter that the comparison is based on independent forensic geometry, not on assumptions about age, and that the two lines of evidence should be evaluated separately rather than forced to agree. Neither side has resolved this tension, and it remains one of the more genuinely unsettled corners of shroud research. One further piece of this story deserves an honest and direct mention, because leaving it out would be a disservice to accurate reporting. In 2018, a research team led by Giulio Fanti and Elvio Carlino published a paper in the journal PLOS ONE Reporting, nanoparticle analysis of shroud fibers that they argued was consistent with severe violent trauma, including possible torture. That paper was formally retracted by the journal in 2024, following a review that raised concerns about the underlying methodology and interpretation of the data. The researchers involved have publicly disputed the retraction and maintain that their broader findings remain valid, and some of the same investigators have since published newer analyses examining additional fiber samples. But intellectual honesty requires stating plainly that this specific line of evidence carries a serious, documented credibility problem, and any account of the shroud's trauma evidence that omits the retraction is not giving you the full picture. Separately from the DNA and blood chemistry work, Physicist Liberato De Caro and colleagues proposed an alternative dating method in the early 2020s, using wide-angle X-ray scattering to measure the natural aging and degradation of the cellulose structure in the linen fibers, comparing it against reference samples of known age. Their results suggested an age range consistent with the 1st century, dramatically older than the 1988 radiocarbon date. This method remains genuinely experimental in the context of textile dating, has not replaced radiocarbon dating as an accepted standard within the wider scientific community, and has drawn criticism regarding sample size and the reliability of comparing structural degradation across textiles with very different storage histories. It is a real, published, peer-reviewed proposal, and it is also, at this stage, a minority position within mainstream physics and archaeometry. Through all of this, the Catholic Church itself has maintained a notably consistent and, frankly, more restrained position than most of the media coverage surrounding it. The Vatican has never issued an official declaration of authenticity. Multiple popes, including John Paul II, Benedict XVI, and Francis, have referred to the shroud publicly as an icon, a devotional object worthy of veneration and reflection, deliberately avoiding language that would assert scientific proof of its identity. The Church has, at the same time, generally permitted continued scientific access to the cloth across nearly a century and a half of testing, including the destructive radiocarbon sampling in 1988, which is not the behavior of an institution trying to hide something from independent verification. Public expositions of the shroud, held periodically in Turin, continue to draw millions of visitors, most of whom describe their visit in terms of personal spiritual reflection, rather than a verdict on a scientific debate. So, where does that leave us? Sitting here in 2026 with a peer-reviewed metagenomic study, a disputed radiocarbon date, a retracted trauma paper, an experimental X-ray dating method, and a companion relic in Spain that keeps turning up compatible forensic details. It leaves us, honestly, without a single clean answer. And that is not a failure of the science. It is what happens when you apply 5 completely independent scientific disciplines: genetics, radiocarbon chemistry, forensic pathology, textile physics, and botany, to an object that has spent hundreds and possibly thousands of years accumulating a genuinely complicated physical history. The 2026 DNA study did not prove the shroud is the burial cloth of Jesus of Nazareth. It also did not prove it is a medieval forgery. What it did, with real scientific rigor, was confirm that this piece of linen has passed through an extraordinary number of hands, environments, and centuries, carrying with it fragments of carrots and coral, 16th century repair thread, the DNA of the modern scientist who first sampled it, and at least one genetic signature that still points tantalizingly and unresolved toward the ancient Near East. Maybe future research will narrow this down further. Non-destructive genetic techniques are improving quickly, and researchers are already discussing less invasive ways to sample the cloth without damaging it. Maybe the debate over the 1988 radiocarbon sample will eventually be settled with new physical evidence rather than competing interpretations of old data. Or maybe, as with so many objects that sit at the exact intersection of history, faith, and physical evidence, this is simply a case where the available science can narrow the possibilities without ever fully closing the file. What we can say with confidence, based on the actual published research rather than the version of the story that spreads fastest online, is that the Shroud of Turin remains one of the most rigorously and repeatedly tested objects in the history of archaeology, and that every time scientists open it up for another look, they find something genuinely new to argue about. If you want to keep following stories like this one, where real peer-reviewed science meets centuries-old mystery, subscribe to the channel and turn on notifications so you don't miss the next deep dive. And if you have your own take on what this new DNA evidence actually proves, or doesn't prove, drop it in the comments, because this is exactly the kind of debate that benefits from more people looking closely at the actual evidence instead of just the headline.